I would like to introduce Dr. Sophia Yohe. She is a professor of pathology in hematopathology service in our laboratory, medicine and pathology department. And she is practicing hematopathology, but along with that, also molecular genetic pathology at the University of Minnesota. She is also the medical director of m-Health Fairview's Molecular Diagnostic Laboratory, and she has spent a lot of time educating herself. This is something you wanted to mention. Educating herself on the FDA final rule, thinking about how to efficiently comply with it, and thinking about the impact on pathology and laboratory medicine. She has been a CLIA director for the CLIA component of the University of Minnesota Genomics Center and the CAP inspector for over 10 years, so she has a lot of experience to share with all of us. So please welcome Dr. Sophia Yohe.
Thanks, Samir. So I know that this is probably not going to be people's favorite topic, since there's a lot of strong feelings about the FDA, but hopefully to keep everyone's attention, I am going to have a couple interactive questions, assuming Poll Everywhere works for me, so you can see the link here. I'll leave it up for a moment so people can pull it up on their phone. I could not figure out how to get the QR code, but I'm going to be talking about the regulatory landscape of laboratory-developed tests. And as medical director of the molecular lab, almost everything we do is a laboratory-developed test. I've been medical director of the flow lab previously, and pretty much everything there is also a laboratory-developed test. So I've thought a lot about how to do this and what the final rule kind of means for us. And that's what a lot of my talk is going to be, although I am going to briefly touch on CLIA and CAP as well. I don't have any financial disclosures. Hopefully people have had a chance to pull that up. There's only going to be a couple questions. But the objectives, not yet. Did everyone get it? Not yet. Okay. All right, well. So the objectives today, I'm going to compare CLIA, CAP, and the FDA final rule a little bit. Then we're going to actually learn about the FDA LDT final rule. And as we go through, we'll talk about what kind of things we already do in our current lab processes that can meet some of the requirements for the final rule and what things are going to be new for us. And that's what we're going to do. Before we start, though, I want to make sure we're all on the same page of what a few definitions are. So the first is, what is an LDT? It is an in vitro diagnostic test that is designed, manufactured, and used in a single laboratory. This is defined by your CLIA certificate, and it is not FDA approved or cleared. It's important to recognize that this includes a significant modification of an FDA approved or cleared test. And we'll talk about significant modifications and what does and doesn't make a significant modification as we go through. I think it helps to understand what the FDA's main beef with LDTs is, and it is that they think LDTs are less safe. And I think it's important to recognize that a lot of clinicians and patients think the same thing. So, I understand our standpoint and where we're coming from, and we don't think that. But I think it's important to recognize that because it can give us some common ground to work with. The other definition I think that's important to know is enforcement discretion. And this is the FDA's choice to not enforce some requirements in certain situations. People sometimes refer to this as being grandfathered, but it's not. And there's an important difference because the FDA can change their mind. They can decide, I am no longer going to have enforcement discretion for this broad thing or for this particular lab. And we'll talk a little bit about that as we go through as well. So first, let's talk about CLIA and what is CLIA's approach to LDTs. So CLIA makes a distinction between an FDA-approved cleared test and an LDT. For an FDA-approved or cleared test, you need to do verification. So there are certain performance aspects that the manufacturer has developed that you have to verify they work in your lab. Those include accuracy, precision, the reportable range, and ensuring that the reference interval is appropriate for your population. Validation is different. These are LDTs. These are also FDA-modified tests. In addition to what you have to do for verification, you have to validate additional performance characteristics. So analytic sensitivity and specificity, including interfering substances, reference intervals, the broad category of other as needed, depending on the test, which could be a lot of different things. And then again, calibration and controls are required for both of these. What about the CAP? What are the CAP's requirements for LDTs? So the main requirements for them for validation is that you include at least 20 samples. These should be both positives and negatives, and it should span the breadth or spectrum that your test is going to do. Again, 20 samples is a minimum. You have to have some sort of clinical claims validation. This is a clinical validity sort of statement. And that can be a full clinical validation where you assess your test and you assess patient outcomes or other things like that, or you can do it based on the peer review literature. Other requirements that we do with CAP is we maintain a list, and that's on file with CAP. Calibration and QC are required, just like in CLIA. And then there are some specific requirements for our reports that we comment that this is an LDT, basically. There's a developed by this lab sort of comment that goes on LDT reports. And you have to have a brief description of the methods, the performance characteristics, and the clinical use. These details can be available on request. They don't have to be in the report, especially if they're quite long. But you do have to have some brief data there. So why does the FDA feel like this isn't enough? The main reasons is that their comment is that a lot of the CAP and CLIA regulation is after the fact. We've done our validation, we have our test in place, and then we do PT, or then we get inspected. There's no evaluation beforehand to make sure that your test is up to par before going into effect. They state that CLIA does not assess clinical validity, and the FDA does, and that is true for CLIA, although CAP does have a requirement for some clinical validity. CLIA is limited to your specific lab situation. FDA is looking more at tests that might be more broad, although they do recognize in the LDT space that these might be part of a single lab, and there are some enforcement discretions that take that into account. The other thing the FDA is concerned about is that there's no monitoring of LDTs. If there's a bad performing LDT, there's no central reporting to identify that. And that actually has to do with one of the first phases in the LDT FDA final rule. So, as you may or may not know, the final rule was published on May 6th. It's important to know this date because all the other dates come from this particular date. There is a phased implementation over 4 years. I've grouped stage 4 and 5 together because they're essentially the same thing. You just have to do higher risk before lower risk. So stage 1, we're already halfway through year 1 for stage 1, is that issue that FDA has that there's no way to monitor LDTs. And what stage 1 is adverse event reporting, monitoring, and at times reporting complaints, and reporting corrections or removals of an LDT. Stage 2, which is year 2, is registration, listing, and labeling. That's what I'm going to focus on. I'm not going to talk about investigational use requirements, but there are some things there if you're doing research that you'll need to know. Stage 3 has a lot of quality systems requirements. Stage 1 has a single quality system requirement, so the rest of the quality system requirements we have to meet is in year 3. And then year 4 is submission to the FDA for the LDTs to get pre-market review, approval, et cetera. And we'll talk about the differences between those because that's also some terminology. And high risks have to be done first, low and moderate risks done in the second half of that fourth year. Let's talk about the areas of enforcement discretion. So I've listed them all here, but I'm only going to go into detail about some of the ones in black. So there's enforcement discretion from all phases, except for for all phases, for 1976 type LDTs, HLA for transplant, law enforcement related forensics only, the VA and the Department of Defense, and public health testing. I'm only going to talk about the 1976 type LDTs because that's what's going to affect most of us unless you happen to work in the VA or the DoD or in public health. There's enforcement discretion for the 3rd through 5th phases for tests that were in use prior to May 6th of 2024, and tests that meet an unmet need within your healthcare system, although there are a few limitations for this. And then also for blood banking, antisera for rare blood antigens, which is kind of a specific thing, and I'm not going to discuss that today. There is enforcement discretion for the last 2 phases, that submitting to the FDA for a pre-market review or approval, if you are New York State certified for your tests, which I'm not going to talk into detail about because that would be a whole talk to talk about New York State certification. And then minor modifications of FDA approved or cleared tests that are performed only within the lab that modified it. And we will talk about what sorts of things are minor modifications and what sorts of things might not be minor modifications when we go through that. Let's talk about 1976 LDTs first. So for those of you who weren't around in 76, here are some of the things that were going on in 1976. And the way the FDA defines these tests is that they have manual techniques. There's no automation. You have specialized laboratory personnel performing them. You use clinical grade components. So these are analyte-specific reagents or ASRs, not research use only or RUOs. And some examples they give is manual staining and manual ANAs. What's the problem with this? This isn't very many tests, right? Most IHC is automated on a stainer. I don't think IHC are going to have enforcement discretion if you use an automated stainer. And given that FDA approval for IHC includes the whole platform, including the stainer, I think that that's a fairly realistic assessment. So I think this is going to really impact a lot of pathology. Previously marketed LDTs. The FDA's main reason for putting enforcement discretion in phases 3 to 5 was to avoid disruption in patient care. So if the patient's been getting a test that's being followed or whatever, that it suddenly isn't available for them. But they are going to use that phase 1 and 2 information that you submit to screen for poor performers. So if they see something that they think might be a poor performer, they have the ability to say, I'm going to check into your lab more. I'm going to make your lab do more because I have enforcement discretion. I'm choosing to back off on that because I've seen a red flag. So my suggestion for your previously marketed LDTs is to make sure that you dot all your I's, cross all your T's with the phase 1 and phase 2 stuff and make sure that that looks good. If you make minor changes to your previously marketed LDTs, you do not need to resubmit or do anything different, but significant modifications do require full submission. So what sorts of things does the FDA consider a significant modification? So a change in indication for testing, a change in the critical reaction components, and significantly different technology. This, I think, does allow for some interpretive leeway, but there are a lot of things that are going to be significant modifications that might alter your previously marketed LDT and then require you to do more. And I think you have to toe the line for this. So one of the things is the clinical indication. This is very important for the FDA. What you say your test can do and what you say your test is for, and having that specific or broad, I think, is a difference, but you also have to be careful about misbranding. One of FDA's concerns for one of the tests on the market, which was non-invasive prenatal testing, which is kind of a standard of care. It's a good test. One of their big problems with it was the marketing that was done that made it seem like more than screening, like it was more of a diagnostic test and not a screen that required follow-up testing afterwards. So misbranding can be an important thing. So I think of this in molecular and I think, well, okay, so what am I going to do with my lung panel or my oncology panel or whatever? You know, if I say that EGFR is being used specifically for lung cancer, and 3 months from now, EGFR becomes important in some other cancer for targeted therapy, I have to change my clinical use statement for that. Can I say that it's for detecting prognostic, diagnostic, and therapeutic mutations in cancer? Would that be considered misbranding? It's broader. It gives me a little more leeway to adjust things. I don't know what the FDA's take on that is, I'll be honest, but I think these are things to consider when you're looking at what am I going to say the clinical use statement or the use of my test is. Now, it's important to remember that we can still use a test outside of what we say our clinical use. So if your clinician calls and says, hey, I really need to do this in this patient for this reason, for us, this is often just a different specimen type that we've not validated or whatever, you can use that. That's part of the medical practice. They're not trying to prevent you from doing medical practice. That would be an exception. You'd have to document that. But you can't advertise it, and that includes internally. So you can't have any internal documentation or advertising that you're doing this sort of off-label use, as it were. Some of the other examples they gave for resubmission is significantly different technology. So adding artificial intelligence or machine learning to a process. One that I don't quite understand, but targeted sequencing to whole genome sequencing. I guess it's a little different, but it's still sequencing. Immunoassay to mass spec, and something going from manual to automated. Anything that adversely changes or has the potential to adversely change the performance or the safety. Another question I think has not been answered at this point is what data is required to support that a change is not critical? Do you have to document your reasoning? Do you have to do some sort of equivalent study showing that it doesn't change anything? What else might need to be done? And I think this is an open question and hasn't been fully answered at this point, but certainly something to keep in mind going forward. The next category is unmet needs testing. So unmet needs testing can only be used in your corporate health system. So for us, that's a problem. Some of our hospitals like Southdale have a different corporate ownership and it would not be considered part of our system for this purpose. Unmet needs testing also has to be a test that does not have an FDA authorized in vitro diagnostic for the condition, or for some reason you can't use that FDA in vitro diagnostic, not because of cost, but because it's not indicated for your patient group or your patient, like it'd be off-label to use it, or the only place that offers that FDA test will not test your patient. They did give examples of rare diseases or sort of rare disease testing like Huntington's and Prader-Willi-Angelman when they talk about unmet needs testing. And in previous versions of the FDA guidance, this unmet needs testing was testing for rare diseases. And it kind of morphed over how it used to be, where they were going to look at a number of tests that were performed over a period of time, and it would be those tests. And I don't like the current form that it came in, because I think there's a lot of problems with it. I think that for rare disease testing that was in place before May 6th, we're probably okay. But for new needs for rare disease testing, I think we're going to have worse test availability and reliability, because instead of having one lab with expertise that can get samples from across the country, every lab's going to have to do their own for maybe doing a few samples a year. And that's problematic because it's expensive to bring these up, to do proficiency testing, to validate, et cetera. Now multiple places have to do that instead of one place. And I think what's going to happen is that people just aren't going to offer some of that testing, and labs will no longer be a safety net for having testing available that's not worth an IVD manufacturer's time to develop. And so for truly rare disease testing that is new, I think this is going to be a problem. And given the FDA's focus on safety, I think this is potentially an area that maybe we could negotiate and get some traction on. All right, so those are the things that have enforcement discretion. Now let's talk about what the actual phases are. Stage 1, which is what you should be working on right now, includes medical device reporting. This is really our serious adverse events reporting. We do serious adverse event type reporting already. You should be tracking serious adverse events that occurred because of your lab test. You should be aware of that. Just realize that there's now criteria for when you have to report that to the FDA. Correction and removal reporting. So if you make a change to your test, you will need to report if it's a significant change. And then the complaints, and we'll talk a little bit more about that. Some complaints have to be reported to the FDA. So the only thing that you are required to report to the FDA is a suspected device-associated death or serious injury, or a malfunction of your assay, I guess, that was likely to lead to death or serious injury. There are other things that they encourage you to report, but they are not required. And those are the only things you have to report, which I don't think we have a lot of death-associated problems with lab tests. But, you know, what is a serious injury? Is a sample mix-up that led to wrong surgery or something like that? That's probably a serious injury, right? That sort of thing would probably need to be reported. Correction and removal. So if you corrected your test because there was a problem with it and it was performed to reduce the risk of giving a wrong result or correct a violation, even if it was an error that caused it, then that has to be reported to the FDA, that correction. Or if you recall a device for the same reason, you would have to report it. You do not have to report withdrawals for other reasons, just because you decide not to offer a test anymore. You don't have to report if you shut down for a while because you have to do equipment repairs or routine servicing or that sort of thing. It's only if it was something that adversely affected the performance of the test that you had to correct. And then lastly, the complaints. So this, I think, will be a little new for us. You know, as laboratories, we certainly receive complaints, but I don't think we have a real standardized way of documenting complaints. And based on this, we now require a standardized approach to complaints. And so you have to think about this. Where are you going to get complaints? Are you going to get them by phone call, by email, by walking down the hall and meeting one of your colleagues? And how do you get that documented? And in addition to documenting, you have to do a timely evaluation of it, document oral complaints upon receipt, and then evaluate the complaint to determine, do you need to investigate it? And if not, why not? If you do, then you have to go through and document some specific things. A lot of them are fairly simple, like who made the complaint, what was the complaint, etc. Do you need to report to the FDA? it is only if it is related to a death or serious injury, or likely to cause death or serious injury. So the same thing that you would do medical device reporting for. And again, if you already reported it under that, you don't have to report it because it also came from a complaint. You only have to report it once. All right, so now we will see if, oh man, it's not showing the whole question. And neither is my phone. Okay, well, we'll see if I can remember exactly what I typed here, but which of the following would require reporting to the FDA? If you update your gene list with the newest HGNC nomenclature, if you realize that you left TP53 off the gene list in error for the last month, and you correct your test to include that, if you receive a complaint from a clinician that a report has an error, you clarify the correct results and correct it, and it didn't lead to any patient harm. If your NGS test identified an EGFR mutation in a lung cancer patient and they don't respond to targeted therapy. And is this working for people? Yep. Okay, yeah, I see stuff. Okay, I don't know how to get this to show. All right, so I am going to, well, I don't know how to complete this. Okay, well, we have most people saying B, and that is the correct answer. So this would be a correction that would require reporting. Updating with standard new nomenclature would be kind of a standard update that you would do. A complaint that does not have a patient safety impact for a serious adverse event or death would not require reporting. You could optionally report it or voluntarily report it, but it would not be required. And D, unless you actually thought that your EGFR test was wrong, you wouldn't have to report that non-response. All right. Let's move on to phase 2. And phase 2 is basically listing your test with the FDA is essentially what phase 2 is. So you have to register with the FDA and list all your tests. This is fairly straightforward. There's a relatively small fee to register with the FDA. We have a list of LDTs already from CAP. This shouldn't be a huge lift for what we do. Labeling requirements, however, are going to be a little bit bigger of a lift, but still not, I think, terrible. It's mostly going to be formatting things correctly. 2 types of labels, product label and package insert. So product label to me makes most sense if I think about it on a drug. Of course, if I think about it on a drug, I have a hard time putting that onto a lab test, but it's the information that you would want that kind of tells you what the test is. So what is the test name? What's the lab that does it? What's the intended use? Any warnings and precautions? I feel like that's more for drugs than it is for lab tests, but some specific requirements, but this doesn't have to be on the product label. It can be in the product insert if there's not enough space. You need some sort of manufacturing history, sort of lot or control number. And one of the new things is going to be a unique device identifier. So you will need to obtain some sort of unique device identifier for your lab test process. And this is usually some sort of barcode that you can get through the FDA, or there are other accredited agencies that can give you UDIs. The other type of labeling is the package insert. And so this is kind of the information that comes with your device or your drug or whatever. And if you look at these things, I'm not going to read these off because it's a long list, but almost all of these are going to be covered in your validation and your protocol. And so it's really just a new formatting of information that we essentially already have. And so again, I don't think this is going to be a huge lift. It's going to be some paperwork, but it's not going to be a huge lift. Stage 3. So stage 3 is where I think there's a few more new things that we're going to have to deal with. And there are several parts. You can see the references here, and I've read all these references. It's very exciting reading, I can tell you that. But design control, purchasing controls, acceptance activities, CAPA, which is an acronym I guess we're going to have to get used to. Those in the manufacturing world know it well, but it was new for me. And then records. Now this is the section that FDA thinks will cost a fair amount of money to put these into place. I actually feel like we have a lot of these in place already, but not all of them. So I'm not sure about that. One of the things I think is a little tough is that they're going to publish new requirements in February of 2026, and when we comply, we have to comply with those new requirements, but it's pretty close to when we have to actually submit. So I'm hoping they give us lead time about what those new requirements are, but you do need to use them. If you do not complete stage 3, you cannot move on to stages 4 and 5. So you do need to complete stage 3 on time. Design control, I think, is the newest concept for what we do. And this is really, I think it's something we do when we really validate or bring up a new test. I don't think it's something we document. And I think this is going to be a big difference. So you have to assess what your user needs are. You have to use that as part of the input for the design of your test. You then have to have a design process where you design something that meets that input and user need. And then you have to look at the output and make sure that it meets your requirements that you had before you kind of have your final LDT. All of this needs to be documented, and it has to be approved as well. So this, I think, is a very new process for us. And I don't think we have a computer system that works very well for this sort of thing either. And you have to keep this in some sort of design history file that documents this whole process. It tracks the inputs to the design and the groups that provided input, the changes that you made to your design to meet these requirements, and that review and approval process. So this, I think, is fairly new for what we in labs do. I don't think this is something we think about so much as having to document this process. All right, one of the other quality systems is purchasing control. So evaluating and selecting suppliers, maintaining a record of suppliers, defining what they do for you, and the purchasing data, including the quality requirements. I think we have these things. We have this with our purchasing departments. We have QC requirements. We do new lot testing, as Dr. Colleen kind of showed. I don't think this is really an issue. It's just making sure that our documentation meets what the FDA's requirements are. I think these processes are already in place. Likewise, I think acceptance activities are mostly in place, where we establish acceptance. What is acceptable performance to say that this test worked? This includes acceptance for incoming products. We already talked about kind of reagent lot comparisons. There's also instrument comparisons. I used, again, an example from molecular for in-process. So if we're doing next generation sequencing, for example, we have several steps along the way to make sure that our process worked. And then in the final, we have batch controls or lot controls or other types of controls to ensure that the entire process worked. And we document these things. Do we have the exact documentation that the FDA wants? Probably not, but I think that we have these processes in place. Corrective and preventive action, which is why they call it CAPA, because that's a bit of a mouthful. And so this is sort of the proactive step. So in addition, it's being proactive and monitoring for quality issues, investigating them and identifying corrective action, and then verifying. I mean, this is kind of our PDSA cycle. And, you know, we monitor for issues through proficiency testing, positivity rates, control trends, comparing with other lab results, etc. Again, I think these are things that we mostly have in place, and it's just a matter of documenting the processes that we do for these corrective and preventative actions for our tests that we have, our LDTs. And the last one in this section is records. And again, we have a lot of records. We maintain records. And our CAP requirements for retaining records and protocols for 2 years after a test is retired is very similar to what the FDA has. I think what is a little bit different here is the organization of the records. So some of our records might be by instrument or other things, whereas the FDA wants a record by test. So for me, that leads to some issues. It's like, well, I use a sequencer for multiple different tests. Can I just link that, or do I have to like copy it in everybody's little spot? So I don't know exactly how that works, but you do have to have a master record for each device. And again, a device is our lab process, which to me is kind of a hard thing to think of a single thing versus a process and try to shove a process into a single thing. But we need that. We need a history record for each device, or a test and test batches, which again, our LIS systems tend to have that information, which instruments you used, which techs worked on it, all of that sort of thing is there. A quality system record, the complaint files, the servicing, and any statistical techniques. So these are all the things that are required in the records. All right, so we have our next question coming up. So which of these processes that we just talked about ensures that a validated LDT meets the user's needs? Are you guys able to see this one, or did I not activate it? Not activated. Okay, here we go. Activate that. Oops, sorry, I hit it twice. There we go. That should now be activated. All right, we don't have a ton of time, so everybody got it right. It is design control. All right, lastly, pre-market review. This is going to be the expensive part. So the FDA divides tests into low risk and high risk, low to medium risk and high risk. Class 1 and 2 devices that are low to medium risk and are not novel, including things that show substantial equivalence to something else that is approved, can go through pre-market notification or 510K. These are referred to as FDA cleared. This is the cheapest version to go through. Pre-market approval is a class 3 or high-risk device, and these are typically novel devices as well. These generally require very in-depth clinical evidence, clinical trials, and these are things that are FDA approved. Now, you can request to get a class 3 high-risk novel device moved into a low to medium risk by adding additional controls to ensure safety and effectiveness. If you do that, that goes into the de novo category, and that is FDA granted when you get approval through the FDA. And that has a cost in between the two. These are not cheap. And I don't know what the best option for this is. The cost differs between whether you're a small manufacturer or a large manufacturer. I have no idea what the FDA is going to consider healthcare systems as large or small manufacturers. But these can be quite expensive. Just a note on substantial equivalence for those low and medium risk for that 510K. You need to have the same intended use and the same technical characteristics, or if you have different technical characteristics, you have to demonstrate that the device is just as safe and as effective as the other device. All right, so we're almost done, because I'm sure that what people are really questioning is, will the final rule stand? So there was a lawsuit started by ACLA, and then AMP joined it. Recently, there was a ruling that actually was for the FDA on this lawsuit. Some people think that the Supreme Court overruling the Chevron doctrine may have some effect. So basically, the Chevron doctrine, the courts have to defer to the regulatory agency. If legislation is incomplete or unclear, the agency gets to interpret it, basically. That overrule means that the court will now determine if an agency's actions are consistent with the statute and intent of Congress. And the basic effect is that there'll be some legal challenges for regulatory agencies to enforce and issue rules if the legislation is incomplete or unclear. There was talk on the House Appropriations Bill about them having some language about taking a step back and readdressing modernization of CLIA. I've spent some time last week reading the House Appropriations Bill, and I couldn't find that language, so I don't know if it got removed or what happened. There is not specific funding for LDT, but there is some discretionary funding that the FDA could potentially use for that. There's also been retirement of several key FDA leaders that have been involved in this LDT. However, my prediction is that at least part of the FDA rule will stand. In other words, we are not going back to the status quo. Again, my opinion, you can have your own opinion. Will we be 100% final rule? I hope not. There are some definite problems with it. I'm hoping that we end up somewhere in between. But no matter what, that somewhere in between is going to take time. If there are additional lawsuits, if new legislation is passed, or other changes are negotiated with the FDA, all of that is going to take time. And in the meantime, you only have until May 6th to get phase 1 in place, which is only about 6 months away now. The FDA final rule is in place until lawsuits or legislation changes it. My suggestion is to get the final rule to a place that's more palatable for pathologists. We need to find areas of common ground where we can focus on changes that address the safety of laboratory-developed tests and the concerns that the FDA and the public has to get those things done. So that's my talk. My references, there's a lot of FDA guidance out there. There's the initial Code of Federal Registrar and what they're basing their rules on. They have some guidance, the final rule itself to read. CLAP and CLIA have resources as well. And there are a lot of webinars and discussions on the FDA final rule by a lot of the pathology societies out there. And I'll take any questions.
Thank you very much, Sophia. We don't have a lot of time, but maybe one quick question, if anybody. Mahmoud? Oh, you don't have? I thought you do.
I left you all speechless.
Yeah, nobody. Yeah, I monitored the online.
Tony.
Tony has one.
The change in administration is significant because
That's true.
a new administration may not be so enthusiastic about FDA regulations in general. And,
Yes, yes, there may be more dismantling of that. That is true. But it's hard to say because